This is the Professor, CBMD PhD. It's today Sunday, November 17, 2024. It is 9:56 and 18 seconds, p.m. Eastern Standard Time. And today's research, Lipid Bilayer. So let's hop to it here. Particles bound to the membrane can move on the surface of a cell. Even integral membrane proteins such as enzymes may move from one cell surface to another. E.g. important example from apical to lateral when barriers to flow such as cell junctions are disrupted. The fluidity of the membrane is a function of the types of phospholipids in the membrane and variations in their local concentration. As previously mentioned, lipid rafts containing integral membrane proteins may move to a different region of the plasma membrane. The movement of an integral protein anchored on a lipid raft makes the signaling more precise and prevents non-specific interactions. The lateral migration of proteins is often limited by physical connections between membrane proteins and intercellular or extracellular structures. Such connections may exist among proteins associated with cytoskeletal elements and portions of a membrane proteins that extend into the adjacent cytoplasm. The cytoplasmic domains of membrane proteins and peripheral proteins associated with the extracellular matrix and the integral membrane proteins that extend from the cell surface. Important example, the extracellular domain. Through these connections, proteins can be localized or restricted to specialized regions of the plasma membrane or can act as transmembrane linkers between intracellular and extracellular filaments. The cell membrane and internal plasma membranes of organelles are continually remodeling. Video microscopy reveals that the plasma membrane undergoes bulging, investigation, budding, tubulation, raffling, fusion, and fission. These processes are essential for virtually all the major functions of the cell, including synthesis and degradation of molecules, endocytosis, exocytosis, signaling, immunologic defense mechanisms, cell division, and migration. In general, there are two types of membrane remodeling. Remodeling that excludes cytoplasm from the lumen of forming vessels or tubules, such as during the formation of endocystic vessels. This process involves proteins residing on the inner cytoplasmic surface of the membrane. Important example, clathrin, cavelins, COPI, and COP2 and others, that help change the shape of the plasma membrane into vessel-like investigation. The domain family of membranes to size and proteins is essential to mediate the liberation of the vessels from the plasma membrane. Similar investigations can be found in skeletal muscle cells, where T-tubules are connected to the external cell environment. In smooth muscle cells, the cavoli and endocystic vessels help regulate Ca2 homeostasis. And this is Professor Christopher, codename Will, leader of research scientist ufologist, computer technician, computer programmer, philanthropist, writer, mathematician, artist, photographer, film producer, chef, animal, and nature lover, passion, world peace. And this video was created for educational purposes only. Hope you enjoyed listening. Until next time, have a great day. This is the Professor, CBMD PhD. It's today Sunday, November